Welcome to the Deep Dive. If you feel like the pace of scientific discovery is accelerating, you are definitely not wrong.
Hey, not at all.
Today, we are strapping into what might be the fastest timeline in all of science. How AI is just collapsing the search space for molecular and genomic discovery.
And it's really not just about prediction anymore, which was already, incredible.
Right.
We're moving so far beyond predicting what exists. We're diving into models that are designing entirely new to nature compounds, whole biological systems, basically from scratch.
But to get why that's so huge, we first have to understand the scale of the old problem, right? The thing AI is solving.
Oh, absolutely. The scale is everything.
Yeah.
For decades, discovery has relied on this brute force approach, high-throughput screening, HTS.
The classic method of just testing millions of things to see what sticks.
Exactly. And the challenge isn't just that the molecular world is incredibly complex. It's that HTS is just so wasteful.
Wasteful how, though?
Think about it. You might test a million different compounds.
Yeah.
Maybe, if you're lucky, a handful work.
Yeah.
So that means 99.9% of all your effort, all that data, is negative data. And that just gets thrown out.
Even though it tells you what doesn't work.
Precisely. It contains all these vital clues, but it's discarded. This leads to this crushing attrition rate, especially for really tough targets like certain immune receptors or protein interfaces.
Okay, so let's unpack that. That's our mission for this deep dive. We're looking for the shortcuts, the AI breakthroughs that just redefine efficiency.
Yep.
We'll be looking at these cutting-edge models that don't just, speed up screening, but fundamentally change how we optimize chemical reactions, design entirely new biological systems.
And even model whole synthetic genomes.
It's all about AI-guided experimentation, delivering results faster than anyone thought possible, even just a couple of years ago.
It's a real journey from brute force lab work to high-level system design.
So let's start right at that pain point you mentioned, the HTS bottleneck. You're hunting for a new drug, you're screening millions of compounds against these really hard targets like the immune receptor TREM2. How does AI tackle that mountain of, well, useless data?
This is really where AI just transforms the whole workflow. It turns a marathon into a sprint. The solution we're seeing is platforms like HTS Oracle.
HTS Oracle, okay.
It's a deep learning system designed for what they call hit prioritization.
Which means it tells you what's actually worth testing.
Exactly. It tells the scientist which 5 compounds out of 1,000 are the ones to focus on. And it works so well because it actually learns from all that negative data that we used to ignore.
And when you look at the performance numbers for HTS Oracle, they're not just helpful, they seem almost mandatory now.
They really do. I mean, for really tough targets like TREM2 and GA3L1, HTS Oracle reduced the number of compounds that needed actual expensive lab testing by more than 99%.
99%.
More than 99%. For another target, CD28, it was a 70% reduction. I mean, if you're running a drug discovery program, that shift in how you use your resources it's revolutionary.
A 99% reduction in physical work is just a massive time saver. But did the quality of the hits also go up? It wasn't just filtering out the junk, was it?
That's the critical point. The predictive accuracy meant the hit rates of the percentage of compounds you test that actually work, it went up substantially.
How substantially?
For TREM2, HTS Oracle got an 87.5 times improvement in the hit rate compared to just, random screening.
That's astonishing. And did this lead to a real tangible molecule in the end?
It did. It successfully prioritized a few small molecules, and one of them, Z2770993-038, showed genuine dose-dependent binding to the CD28 protein in the lab.
Okay, you're going to have to translate dose-dependent binding for
It
it.
just means the more the molecule you add, the more it sticks to its target, which is what you want to see. Its binding strength, the kV value, was in the low micromolar range, about 43.35 micromolar.
Right. And in plain English, a KD of 43.35 micromolar means what?
It's a measure of how tightly it sticks, lower is better. So getting a value like that from an initial screen on a difficult target, it proves the AI found a real functional starting point, a major win.
Okay, so we've solved the screening problem. We can find the needle in the haystack almost instantly. But now you have to make that needle. Chemical synthesis itself is often a black box, right? Starved for good data. Is AI optimizing the actual chemistry, too?
It really is. We're seeing AI take over the lab bench, so to speak. This moves us from finding hits to optimizing the reaction. And a great example is a system called ChemBOMAS.
ChemBOMAS?
Think of it like an intelligent robotic chemist. It's built on an LLM-enhanced multi-agent system.
Whoa, that sounds incredibly complex. An LLM running a chemistry lab. How does it even begin to manage all the variables in a reaction? temperature, catalyst, solvent.
It's smart. It breaks the problem down into 2 stages. The first stage is what they call knowledge-driven coarse-grained optimization. The LLM basically acts like the world's best research assistant. It scans all the chemical literature, databases, and it uses real physicochemical properties to rank the parameters like which catalyst is most important, which solvent matters less, and that narrows down the promising areas to search.
So it's using existing chemical theory to tell the system, hey, don't even bother looking over here. The yield is going to be terrible.
Precisely. It constrains the search space using all of accumulated human knowledge. Then comes stage 2, data-driven fine-grained optimization.
Right.
Once the search is narrowed down, the LLM generates what they call pseudo-data points. These help accelerate a statistical method called Bayesian optimization, which lets the system run tons of digital experiments very quickly inside those promising zones.
Wait, I have to stop you there. Pseudodata. How do we know the AI isn't just hallucinating a perfect but completely impossible 96% yield?
That is a fantastic question, and it's why the 2-stage approach is so brilliant. The system only accelerates the search within the chemically plausible regions that were defined by all that literature in stage 1.
So it's grounded in reality.
It is, and the real-world validation proves it. They tested ChemBOMAS on a really challenging, previously unreported palladium-catalyzed cross-coupling reaction. Super complex. 6 different parameters to adjust, all under tight constraints, like low catalyst levels.
And the results. How did it do against a human expert?
In that tough 6-dimensional space, ChemBOMAS achieved an optimal product yield of 96%.
96.
Wow. Now compare that to the 15% yield that human domain experts got under the exact same constraints.
That's more than a 6-fold improvement. It feels like a paradigm shift. We've gone from finding molecules to making them super efficiently. Let's pivot to the next frontier, creating molecules that don't even exist in nature.
This is where we get into de novo generative design. This is the ability to engineer structure and sequence completely from scratch. You can create brand new proteins, peptides, anything to do a very specific job.
An inhibitor, a diagnostic tool.
An activator, you name it.
To do that, the AI has to have this intuitive grasp of physics, right? Of how atoms and sequences actually fold up. What are the big tools for that?
BoltzGen is a prime example. It's an all-atom generative model that actually unifies the design process with structure prediction. It can handle all sorts of things, nanobodies, peptides, and can go after almost any target. It's a very general platform.
And the real test of any generative model is generalization, right? Can it solve a problem it's never seen before?
Exactly. And this is where it gets really compelling. They tested BoltzGen on 9 totally novel targets. These were proteins with less than 30% sequence similarity to anything in the known database, the BDB.
So they were completely new problems.
Totally new. And BoltzGen successfully designed nanomolar affinity binders for 66% of them, 2/3.
Nanomolar affinity that's fantastic. That means it sticks really, really tightly.
Incredibly tightly. So the AI didn't just copy something it had seen before. It solved novel structural problems and delivered highly functional binders on its first attempt.
Did it do more than just bind? Were these functional?
Oh, yes. It generated specific functions. For example, they had BoltzGen design these inhibitory peptides, 10 to 50 amino acids long, to target an essential bacterial protein called gyra.
Which is critical for bacteria.
It's critical for them to replicate their DNA. The designed peptides basically act as molecular monkey wrenches. They disrupt the gyra-gyra interaction, and the result was, well, in some tests, the effect was so strong, it completely eliminated the bacterial cell population.
That's pure functional de novo invention. Incredible.
It is.
And speaking of invention, you mentioned this idea of cross-modality design proteins, RNA, DNA, small molecules, all in one system. That sounds like creating a universal language model for science.
That's basically what ODesign is. It's being called the first of its kind generative world model for this kind of cross-modality design.
How does it work?
Its core innovation is a unified token space. Essentially, it treats the minimal chemical units of all these different molecules, amino acids, nucleotides, organic compounds, as if they're all part of a common alphabet.
So instead of having one language model for English and one for French, ODesign has a unified alphabet that lets it become fluent across proteins, DNA, and RNA.
That's a perfect analogy.
Yeah.
And the performance boost is significant. On tough tasks, like designing RNA monomers, ODesign had nearly twice the success rate of a model that was trained only on RNA.
Wow.
It suggests that a unified understanding of biology and chemistry leads to better, more generalized design.
Okay, that's design. But what if a lab has a protein that already works, but it's just too fragile? It falls apart in heat or acid. Can AI help with that, with improving specific properties?
That brings us to Prime. It's a deep learning model that tackles protein optimization using something called temperature-aware language modeling. It's learned not just the sequence of proteins, but how that sequence dictates its stability under different conditions.
And the most impressive thing is its zero-shot capability, isn't it?
Yes, zero shot is a crucial detail here. Prime can suggest mutants with better stability or activity without needing any prior experimental data for that specific protein. A scientist can just feed it a new sequence, and it instantly suggests the best tweaks.
Can you give us a real-world example?
Sure. It was used to enhance the thermal stability of LBCAS12A, which is a key enzyme in some CRISPR systems, making it much more robust. It also improved the resilience of a nanoantibody, a VHH, to survive in extremely alkaline conditions.
And how often did its suggestions work?
Across all the tests, more than 30% of the single-point mutants that Prime recommended showed superior performance to the original natural version.
Okay, we've talked a lot about static structure and sequence, but we know the real story of a protein isn't static. They're dynamic, moving things. Things like binding energy depend on their dynamics, the whole range of possible shapes they can take.
You've hit on the major limitation of static models. A picture is only half the story. You need the whole movie of its movement. The problem is, conventional molecular dynamics simulations that model this, they are so computationally expensive.
Days or weeks on a supercomputer.
Exactly.
So how does AI speed up the movie?
That's where a model like DeepJump comes in. It's a generative model that uses techniques like flow matching and specialized neural networks to map the transition path from one protein shape to another, but it does it almost instantly.
So instead of calculating every tiny step, it just learns the shortcut.
That's a great way to put it. And the result is orders of magnitude speed up compared to conventional simulations. And the most impressive part is that it's trained on very short clips of movement, but it can generalize from those to accurately reproduce the long-term complex dynamics.
It's predicting the whole movie from just a few frames.
That's right.
That's the atomic level. Let's pivot one last time to the most monumental scale, entire genomes. We're talking about training language models on the code of life itself.
This is Evo 2. It is a truly generalist model. We're talking 40 billion parameters, which puts it in the same league as the big commercial LLMs. But it was trained on genetic sequences from all domains of life, over 8.8 trillion nucleotides.
It's like it read the entire library of life's instruction manuals.
That's basically it.
So what can a model with that kind of universal biological knowledge actually do?
It's an incredible generalist predictor. It can accurately predict the effects of mutations on protein function, on an organism's fitness, and it achieves state-of-the-art performance in predicting pathogenicity, whether a mutation causes disease.
And it's not just simple mutations, right?
No, this is key. It's great with indels insertions or deletions in non-coding variants. These are incredibly subtle changes that traditional models really struggle with, but they're often the cause of complex human diseases. Evo 2's performance, validated against clinical data like ClinVar, is a huge step for personalized medicine.
And on the generative side, it's not just predicting small changes, it's building new blueprints.
It is capable of genome-length sequence design. Evo 2 can generate synthetic sequences that have the same length and organization, the sitting as a whole, human mitochondrial genome or a minimal bacterial genome. It's not just editing code anymore. It's writing new operating systems for life.
Okay, let's zoom out and synthesize this. We've seen AI move from a helpful lab assistant to basically a chief architect. In just this short deep dive, we've covered systems that cut experimental screening by over 99%.
That's HTS Oracle.
To pure generative design, creating high-affinity binders for completely novel targets with BoltzGen. We're modeling molecular movement orders of magnitude faster with DeepJump.
And designing life from the atom all the way up to the whole genome with Evo 2.
Yeah.
I think the overarching theme here is the integration of reasoning and generative modeling. We're moving away from these siloed prediction models to fully integrated end-to-end design platforms. High-level reasoning and structure generation are merging, and this fundamentally shifts the scientist's role.
Away from brute force screening.
Away from that, and toward high-level hypothesis generation. System-level orchestration. The machine is designing the blueprint. The entire definition of biological research is being rewritten. We're rapidly approaching a point where the in silico design, the digital creation part, is the most reliable and maybe even the easiest to step in the whole process.
Which raises an important question for you to think about as we wrap this up.
Yeah.
If AI can now reliably design the optimal sequence and structure for new proteins and small molecules for almost any target, how quickly will the remaining bottlenecks, things like manufacturing at scale, production, and effective delivery systems, how quickly will they become the exclusive focus for the next generation of scientific accelerators?